This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 628, recorded on June 17th, 2020. I'm Vincent Drackaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello, Vincent. It's, I just realized that I probably spent 15 minutes yakking at you in the pre-show. Maybe there ought to be a 15-minute Easter egg.
It's good stuff.
It's 89 degrees here in Austin, Texas, and sunny. And I wouldn't have known that because we went out for a nice long walk early. But since then, I've been inside cramming for TWiV. And we're headed for, says 94. Which, maybe I'm becoming, I wouldn't say a Texan, but at least acclimated. 94 here in Texas with relatively low humidity doesn't bother me. It's okay.
Well, it's 26C and partly cloudy here. And it's a nice day. Mostly I've been inside all day. We were talking in the pre-show about what is going on around Austin and terms of infections and how they've been going up since, it looks like 2 weeks after Memorial Day, they started rising, right?
Yep. And I was pointing out this, we might even stick this website in the show notes, the Austin Department of Public Health. They've had a website that you can connect to. This is through austintexas.gov that you can connect to for a long time, but I haven't looked at it for a long time. And it has gone through a very sophisticated evolution with all sorts of data on it. One of the most interesting is this graph that shows a moving average of 7-day average of hospital admissions in the metropolitan statistical area around Austin. It's a 5-county area, and it shows that, about 2 weeks after Memorial Day, this starts to shoot up and is increasing to put us above a 7-day running average of 20 cases per day, which puts us in what they call stage 4, which they have descriptions for how we should behave at various stages. The descriptions are even parsed out as whether you're, at risk, like being old, or other comorbidities. I guess old is a comorbidity. Or if you are, not in one of those categories, what the differences in behaviors might be.
Well, the problem is that the virus never went anywhere. It's still out there.
Well, yeah. And in fact, in my mind, this is a different situation in that if you want to call it first wave, I don't even think of this stuff as waves because I think it's just, this is all one big wave as far as I can tell. Yeah, in some cases, the lockdown managed to get it under control and things decreased. If you look at the situation in Texas, it never really leveled off, which is one of the reasons the governor was criticized for opening up the state because there was never really leveled off, but I won't try and evaluate whether it was better or worse here than in other areas. But to me, initially, part of what was going on is the virus spreading from one place to another. My picture of it now is it's everywhere. Okay. And so as we lift restrictions, it's going to, come out of the ground and start infecting people. So some way or another, we have to learn how to live with this.
Some people have asked, where is it? And it's in people who are mildly or asymptomatically infected.
Right.
It's just spreading silently, and that's where it is.
Yeah.
And I agree. I think I'm coming to the realization now that we can't close down again. I think that's right. I think we have to take precautions like face masks. Here in the New York City area, everybody pretty much wears face masks. And wherever I go, I wear a face mask. And I think if everybody did this, this would really help a huge amount, but not everyone is doing it. I see people still without face masks.
And that's just part of the thing. That is representative of a collection of behaviors. Face mask, social distancing, hand washing, just a consciousness of the fact that this virus is out there and how it's transmitted, and what you can do to minimize the transmission. And beyond that, we have to learn how to live with it. And and by saying that, we can't shut down again, I don't know if some locality became completely overwhelmed again, they may have to shut down. I also don't think that shutting down initially was any kind of mistake or anything. In some places where they were completely overwhelmed, that was obviously the thing to do. But also, we didn't know what was going on. We didn't know what to expect, and so shutting down everywhere at the time was, I think, an appropriate thing to do. We know a lot more now, and I think we know, as you say, that we're going to have to learn how to deal with this. And so, wear a mask, wash your hands, keep your distance.
Here, what I think is that during the shutdown, we were supposed to do catch up, like hospitals figure out how to take care of patients, ramp up their capacity. We get face masks storage, we get stocks of face masks, and most importantly, we ramp up testing. Unfortunately, that has not really happened. And I think that's the problem here, is that without extensive Tetris, these outbreaks are going to spiral. And we had time to do it. And you know what I think now? We didn't do it. We're going to pay the consequences. It sounds pretty harsh, but I don't think the country can shut down again. It'll destroy it, really. We have to learn how to deal with it. There are ways to deal with it, and other countries have, as we've heard in emails, right?
Yep.
All right, let's.
And actually, we were also talking pre-show. There was an editorial in our paper today, and this is what we've been saying: is that sure, you can legislate a lot of this of appropriate behavior and that kind of stuff, but in the end, it's on us as individual citizens to try and understand what's going on and behave appropriately. Mm-hmm. Understand what the risky behaviors are. Understand how you can be safe and behave appropriately. There's another good, I don't know how well documented it is, but a chart going around that I'll look up later that compares different activities that you could be involved in, like exercising outside on one extreme, or at the other extreme, going to a crowded bar all night, and how risky these activities are. And, it's something to think about. Are you going to engage in risky activities? And not just, the consideration is not just whether you're going to be affected, but how you're going to affect your community. Okay.
Yeah, it is on you, but not everyone's well informed. That's the problem too, right?
Well, I was thinking about that today too, because I had to dig around to find all this stuff, and I was thinking, geez, the newspaper ought to run. The newspaper's saying, okay, so we're going to stage 4 in Austin, and I'm thinking, what's stage 4? And I had to dig around through this site to find all that. I don't know where people get their information. I'm a data junkie. So I'm looking for this, but the paper ought to run a feature article, but who's that going to reach? That's even not going to reach a majority of the population. I don't know how you get to everybody and educate them. We'll do the best we can, but my guess is that probably TWiV is not going to reach the largest percentage of people in Austin.
We hardly reach anyone in the scheme of things, that's for sure.
Yeah.
Nope. We're still a niche thing, really.
Yeah.
And nevertheless, we are going to tackle a bunch of your questions today. Before
we start, I want to do one little thing.
Yep.
I want to give a shout out to Devlin.
Mm-hmm.
Okay. Devlin, you know who you are. He's graduating from high school. I think when's this episode going to drop? Tomorrow?
Yeah.
Okay. So when you're hearing this, Devlin, if you're listening to it on the day it drops, it ought to be Thursday. That would be June 18th. Congratulations on your graduation from high school. Keep listening and enjoy your life.
All right. If you like what we do, consider supporting us, microbe.tv/contribute. A couple of scientific reports before we get into your questions. The first, a preprint. I call them scientific reports. I don't want to call them papers because they're not published yet. This one's a preprint from a group at the Scripps Clinic, Scripps Research Institute, and it is called the D614 mutation in SARS-CoV-2 spike protein reduces S1 shedding and increases infectivity. First problem, D614G is not a mutation. It's an amino acid change. I know I'm being pedantic. Mutations are in nucleic acids, not proteins. And these are scientists. They should know better. In any case, this change, D614G, has been kicked around for a couple of weeks now. Ever since a preprint came out, we talked about saying that this spike change has emerged and propagated through many parts of the world. And based on just that, they concluded it was increasing transmission. And of course, the press had a field day with this, and we informed you here on TWiV that it is only an observation. To prove that it increases transmission, you need to do some experiments. And that's really hard to do because they would have to be done in people or at best an animal model where the transmission occurs. And so far we don't have any. I guess this is the ferret model. They transmit, but really poorly.
Actually, I found this, another paper that we're going to do today refers to another paper that I put in the show notes. Maybe we can talk about this briefly, that uses the Syrian hamster as a model. And it's not a great pathogenesis model because they don't get real sick. They lose some weight. They don't have many other, if any, signs other than in postmortem. Am I using that word correctly?
Signs. Perfect.
Okay. I'm learning. And it's over for them really quick. But if you either co-house animals or put them in adjacent cages, they transmit, apparently. I'd like to, it's not a large number of animals. The co-housing and stuff was done 24 hours after the initial infection. So I worry a little about whether it's real transmission or there's just still inoculum floating around. But at any rate, it's if that's true, it looks like a robust transmission model, even too robust for this, because all of the animals get it. You would want a model where you get maybe 25 or 50% transmission, and then see if this virus would cause more.
Yeah. Because,
by the way, this Scripps Institute is Scripps, Florida.
That's right. In people, apparently, 80% of the transmission is done by 10% of the infected people. So that's not what this model is doing, but it's better than nothing. So in this paper, they ask, what effect does this amino acid change? It's in the spike glycoprotein. What does it do in cell culture? And they make pseudotyped viruses. They make retroviruses with the spike of SARS-CoV-2 with or without this change. They even make pseudoviruses with all of the structural proteins of SARS-CoV-2 and the rest from this retrovirus. They make another kind of pseudovirus. I don't remember what. Well,
I saw a pseudovirus that was a murine leukemia virus pseudotyped with the S protein, and then virus-like particles.
Okay, empty. Okay? No genomes. Got it.
I don't think that had a genome. I'm not.
That's right. Because all this is not done in a BSL-3. They wanted to get it out quickly. They might not have a BSL-3, whatever. And they say that with the one amino acid change, they say these pseudotyped retroviruses infect cells more efficiently And this correlates with more incorporation of the spike into the virus particles. Same thing with these virus-like particles, to which I will say, and then they conclude, these results show that this altered spike is more stable than the wild type, consistent with epidemiological data suggesting that viruses with it transmit more efficiently. So, as you may remember, the data don't really prove that they transmit. It could be that this mutation was simply established early on, and therefore everyone else gets infected with viruses with that change. That's called a founder mutation. We just don't know. So I don't think that you should say it transmits more efficiently, but I would say that these experiments done in cell culture with pseudotype viruses and in cells that are completely irrelevant, they're a transformed human tumor cell line called 293Ts, don't tell us anything about transmission in people. They tell you that the change gives you more spike in the particle, and in this particular system, more efficient infection. But my view, it really doesn't tell us anything about transmission, yet the press has been going crazy with this. And they have been quoting virologists saying that this is important, and I just don't think it is. And I think these data are interesting, but I don't think they prove anything about human transmission. And that's my 2 cents.
So going back to that, first paper that was discussed, and you and Nels discussed it in detail,
right,
on TWiV, and I was ambivalent about this. Mm-hmm. Okay, it does appear from the epidemiologic data that this mutant has over time increased in fraction of infections. I don't know how to say this. The fraction of viruses out there, it seems to be increasing with this particular genotype. They claim in that first paper that this particular, and this to me is key, that this particular mutation was positively selected.
That's right.
That's a really loaded term. Okay. And it refers to, I would say, computational biology techniques that you can use to look to see whether a particular genotype is actually undergoing positive selection in the environment. And even after that TWiV that you guys had, I was feeling conflicted about this. I didn't know whether that was true or not. And I called Nels and I said, they say in this paper that they've shown that this is positively selected. What do you think? And Nels says he didn't think they'd proven that. Okay. So to me, that's critical. I want to see independent studies or some other computational biologist weigh in. I can't do it myself. I don't know how to evaluate the data to say whether or not this mutation was positively selected. Beyond that, I think, everything you say is true. This is pseudotype virions, it's cell culture, and et cetera. It is interesting that this particular mutation results in two things, really. More of the S protein incorporated into particles, I will call them, in these two different systems, the virus-like particles and the pseudotype particles. I find that interesting. And also, we've talked about this before, the spike protein is processed into two peptides, one of which is S1, responsible for receptor binding, and the other ultimately responsible for fusion. And this mutation sits at the interface between those two in the cleaved protein. And at least in this paper, I don't know so much about in the virus, but apparently the particles can shed the S1 that is not actually anchored in the membrane, but associated with S2 after the cleavage. And in particles bearing this this particular protein, variant protein, a smaller fraction of the S1 is shed into away from the particles. So there's more S1 on the particles. That would that's the receptor binding domain. So the fact that there's more of this on the particles and more of the S1 contained on what protein there is would seem to correlate with this hypothesis, if you like, that this would transmit better.
Right.
Let's call it a correlation.
The other aspect of this is if this change is being positively selected, there has to be selection pressure for that. All right. And the idea is that it's making it the virus more transmissible. But it seems to me it was pretty damn transmissible from day one. And so where is the selection for that? No one has talked about that, but it's doing really well without it. This change is not in every isolate from every person in the world. Okay, so it doesn't make sense either. I mean, in the end, we may not be able to sort this out because you can't do an experiment to see if it's more transmissible in people. Maybe you could use this golden hamster model that you referred to, right?
Do you know how prevalent this particular mutation is in the U.S.? I remember seeing it at gaining dominance in Europe, but I forget what the U.S. prevalence.
Yeah,
was.
I don't recall, but it's not huge for sure. Now, in contrast, and this is a very different outbreak, but SARS-1, about halfway through the outbreak, a mutant arose, which then was the only virus isolated for the rest of the outbreak. And that was clearly positively selected and was thought to actually reduce the virulence of the virus, although that's hard to show also. But it's hard because you can't do the right experiment. Now, if you remember, a couple of years ago, we had a similar situation with Ebola virus, West African outbreak, a spike amino acid change arose early on that propagated throughout the outbreak. All the subsequent isolates were of that. So it's a bit different. Cell culture models showed more efficient infection, but in animals, it seemed to be attenuated. So in the end, we have no resolution of that either. So these things are very difficult to sort out. And I guess the only thing I want to say is that it's inappropriate for the press to conclude that this is changing the properties of the virus in people. That's really what I think is inappropriate. Normally, this would be a discussion confined to virologists and evolutionary people, but because of the pandemic, it's spilled over into the public, and they're giving a false impression of what's going on here.
The other thing is that nobody is saying that this virus is somehow more pathogenic.
No, thank goodness.
Okay, there's a distinction between transmissibility and pathogenesis. In theory, it seems to me, you could have a virus that transmitted better from one person to another, but in any given individual is not altered relative, shows no difference in pathogenicity. Okay. And nobody's claiming that this causes any more severe disease or is more pathogenic. And there are no data on that one way or another.
Right. So the hamster paper is in Nature, right?
Yeah. Let's see here.
Pathogenesis and transmission of SARS-CoV-2 in golden hamsters.
I just looked at this briefly. I looked at it because it was referred to in this other paper that we're going to discuss.
So they have an intranasal inoculation. They find respiratory tract multiplication and pathology, and then rapid viral clearance by 7 days. They also find some virus in intestinal epithelial cells. It transmitted, right, by air, by direct contact and by droplets. They
do 3 different experiments. They either co-house hamsters, and in this case, it's 1-to-1. They got one inoculated hamster and one hamster co-housed, and they have 3 groups of those in each of these experiments. So it's not a huge number of animals, okay, but it's what results they have are consistent. So one experiment is to actually co-house the animals. And in that case, all 3 inoculate. Oh, and they inoculate a hamster and put him in the cage, co-house him a day later. Okay. 24 hours later. And this infection sweeps through these animals really quickly. It's just a few days. So, and that 24 hours is kind of when they're peaking. The inoculated animal is peaking with respect to virus load. And as I said before, signs wise, you can't tell much. Maybe they ought to put these hamsters in their wheels.
That's
right?
Great. Yes.
And see what they do.
Yeah.
I love that as an asset.
That's very good.
At any rate, they lose some weight. My guess is they would not be exercising as much. At any rate, if you co-house them, they get 100% of the 3 animals transmitting to 3 other animals. Then they put them in adjacent cages, I assume spaced in a fashion where the animals can't actually touch each other. And in that case, they get, once again, 100%. So theoretically, that's aerosol. And then the other thing they do is what they call fomite transmission, where they put the inoculated animal in a cage for a while and then take them out and put a naive animal in the same cage. And in that case, they see transmission in 1 out of the 3 animals. That's not as efficient. Interesting study, I thought.
Now, one of the key points also is that the infectious virus ends at day 7, but they can find RNA for 14 days. And
in these animals.
Yes, and they say the communicable period correlates with the detection of infectious virus, but not viral RNA. And that's important because this is what we're struggling with in people who continue to be PCR positive for weeks. The question is, can you send them home? Can you send them somewhere else? And we're not sure yet because not enough infectivity studies have been done.
And we'll get to that. I want to make sure we properly attribute these papers. The first one we talked about the D614G change, has as co-contributing or equal contributing first authors Zhang, Jackson, and Mu, and as senior authors that are both listed as corresponding authors, Farzan and Choi. And there are other authors on the papers, Oja, Rangarajan, and Izzard. And by the way, Cody Jackson was, I think, an undergraduate at UF. I was familiar with him when he was doing a master's in McFadden's lab. Okay. And he's obviously got himself a gig here. And the Nature paper is actually peer reviewed.
Yeah.
But it's a preview. And that first author is Xia, last author Yen, and that's out of China. I don't know exactly where.
Hong Kong.
Hong Kong?
Yep.
There you go.
Right. One more. And this one is a meta-analysis. It's a meta-archive preprint. Shedding of infectious virus in hospitalized patients with COVID-19, duration and key determinants. First author Van Kampen, last author Van der Eek, and this comes out of the Netherlands.
Actually, it's more than a meta-analysis. It starts off with a meta-analysis, and then they do their own, data collection on 219 patients.
Yeah, that's right. But they don't do any actual clinical study, right?
Well, the way I read it, yeah, they do. They got 219 patients.
Yeah, they did. That's true.
So they start off. It's easy to confuse it as a meta-analysis because they start off doing a meta-analysis and saying that their meta-analysis leads them to conclude
that
there aren't enough studies. Okay, that nobody has correlated RNA, serum antibody, infectious virus, and disease symptomology. Right. And that's one of their biggest gripes, is that the meta-analysis that's been done before has been done primarily on mild cases.
That's right.
Not the meta-analysis says that the studies that have been done before correlate RNA and virus mostly in mild cases, and there aren't many of them. Yeah. So they wanted to do many more, and they wanted to include severe cases because this addresses this problem that we've been talking about, is how do you know when to release somebody from the hospital out into the population, and what precautions do you tell them to take? The basic question being, are they shedding virus?
That's
and the
right.
problem is that in order to really understand whether somebody's shedding virus, you have to take a sample and do a plaque assay.
That's right.
Okay. And that's BSL-3, and it's not easy.
Mm-hmm. Right. So 129 COVID-19 patients, all hospitalized.
129. Okay.
89 intensive care, 40 medium care. And then they looked for these things Rich said, RNA, infectious virus, serum antibody symptoms. And here's what they find. Infectious virus shedding found in 23 out of the 129 patients, 17%. Median duration of shedding, 8 days post onset of symptoms. Okay. And the probability of detecting infectious virus dropped below 5% after 15 days post onset. Viral loads above 7 logs of RNA copies per mil associated with isolation of infectious virus. So in other words, you have to look at the COT value for your PCR, because if it's high, that could indicate infectious virus. And finally, serum neutralizing antibody titer of at least 1 to 20 was independently associated with non-infectious virus. So in these patients, mild, severe patients, shedding doesn't last a long time. And this is upper tract shedding, nasopharyngeal shedding. It doesn't last a long time, 8 to 15 days post symptoms. And if you have high antibody titers, that correlates with dropping of virus.
So a couple of observations. They have some patients that are where they can detect active virus as long as 20 days after the onset of symptoms, which is longer than has been previously reported. We got people who show up RNA positive for a long time, but this is a study of active virus. And so that's longer than previously reported. And the other thing the big question I have about this is, how sensitive is the plaque assay? Okay. Because there are a lot of people, and they only got, you have a whole bunch of people who are RNA positive in the hospital with symptoms at the peak of their infection. And they have less than 7 logs of RNA. You cannot detect infectious virus in them. And I have to wonder whether that's because the cutoff for the infectious virus assay just is too high.
Yeah, it might be.
Or the sampling or the assay itself is just not efficient enough. Because I have a hard time, this is a bias, it's a prejudice, but I have a hard time believing that people who are at the peak of infection and feeling really sick and have a positive PCR test, even if it's quantitatively not that much RNA, it's a little hard for me to believe that like 70% of these people don't have active virus. I don't know.
Yeah, it's true. It's a good point. We don't really know how sensitive it is. It may be that it's not the right cell type. Who knows? People should tinker with it.
But the other correlation is that it's clear that as the quantitatively the amount of RNA drops off and serum antibodies go up, the ability to detect active virus decreases.
That's right.
So what they're saying is that in order to effectively evaluate people, you really need to look not just as plus or minus whether or not they're PCR positive, but quantitatively, how much RNA do they have and whether or not they have antibodies.
That's right.
We're trying without a plaque assay to evaluate whether people are safe to leave the hospital and go home.
Yeah. Well, they're saying that these results warrant the use of RNA and serology to discontinue or de-escalate infection control. So I think, they should need to do more because this is only 129 patients, but I think it's a good point. The other thing I was thinking about the plaque assay, it could be that there are a lot of defective particles in these preparations or in these isolations from patients, right? So you have a lot of RNA, but not a lot of infectivity, and that could be looked at, right?
Yep. Yeah, actually, as I read this article, I don't think it would work. But what comes to my mind is I want to see electron microscopy on all these samples. Okay, and see if I can actually see particles. But there are other methods. You ought to be able to do some sort of fluorescence assays. I mean, there are fluorescence assays that allow you to look at particles. Yeah. Maybe the samples are just too dirty to do that and full of debris and stuff. So maybe the background would be too high.
I think a key here too is that, shedding does not go on for months. 18, 20 days seems to be the maximum that we're seeing. Maybe you get RNA for longer, but it doesn't matter. That's the picture emerging.
Just blanked on what I was going to say. It's okay. It'll come back to me.
All right. And finally, all right, that's the last paper. I found a site which is interesting. I just want to put up there. It's called the COVID-19 Host Genetics Initiative. And it's basically trying to figure out the genetic determinants, host determinants of susceptibility to severe disease and outcomes. And so it's a place for collecting data, right? So this is an area to share resources, organize them, and make a platform for analysis. So people who are working on this can come here and contribute, and you can look at it if you'd like, because the results are here as well. So I think that's pretty cool.
So my understanding of this, correct me if I'm wrong, was that the idea is to collect data on genotypes of individuals.
Yes.
Who respond and are infected or not and respond in one way or another. So my first reaction to this is that I have seen kicking around on social media. I've never seen an article. The idea that blood type makes a difference in outcome from the infection. And I've never seen a scientific report on this, so I don't know whether there's any association, but it seems to me that this kind of site could collect that kind of data.
Yes, exactly. It will collect genotype and disease severity, and then you can try and make correlations, right? The problem with the blood type, there seems to be an association with a certain blood type, type A, I believe, with milder
A positive.
disease.
That's me, dude.
Milder disease. But the problem is they say near the loci for the blood type, there are other immune response genes, which could also be part of that, right?
Right.
So you can't dissect them unless you do a more powered study, and that's what they're working on. But yes.
Yeah, and maybe that'll come out of this kind of analysis.
Hopefully.
By the way, I remember what I was going to, the other thing I was going to say on that previous paper is that another thing that we don't know is what the infectious dose of this virus is. Okay. So if a person is shedding a certain amount of virus, you don't really know what the consequences of that are when they're out in the population. You don't know how much of a virus load really makes a person a threat out in the population. I'm sure there's all sorts of other things that affect that, but I don't know how much virus I need to take up by what route in order to become sick. Is it one particle? Is it 1,000? Do I have to be around somebody who's really, really sick? Well, obviously not, because there's asymptomatic transmission.
That's right. Well, how would you figure that out, Rich?
I don't know. Infect a bunch of people? I mean, if we're going to, I'm joking here, but if we're going to do challenge studies, we may as well do a dose response while we're at it in the placebo group.
Are you volunteering?
No.
Yeah, I didn't think so. And the problem is, if you do a challenge study and you pick young people who are healthy and not likely to die, you're not knowing everything.
Well, yeah. And the other thing about that, if I understand it correctly, correct me if I'm wrong, but usually the licensure of a vaccine
that's right.
is tightly coupled to who the trials were done on.
Exactly.
That's why they have old guys like me in most of these other trials, because that's a susceptible population that you want to be able to vaccinate once it's licensed. Right. If the challenge studies are all done in 20 and 30-year-olds, then that's if for the licensure, it's going to leave me out of it. And we're the ones, the really comorbid ones, who are the best candidates for a vaccine.
That's right. It's a flaw too. You're absolutely right. All right, let's do some email. Bill writes, as far as Melville-inspired cat names go, so someone last time had Ahab.
Yes. Dixon said he had a cat named Ahab that died of, this is relevant, it died of feline infectious peritonitis.
Right. That's right.
Okay. So this is all virology, dudes. Okay. And I commented that Ahab was a cool name for a cat.
All right. So this Bill writes, as far as Melville-inspired cat names go, I've always been partial to Bartleby. For much like the titular character of Melville's Bartleby, the Scrivener, most cats would prefer not to do much that's asked of them. It's true. Thank you for everything you do to keep your listeners informed. Just a humanities major.
Just in quotes. So Bill's listening closely.
That's right.
Good for him.
All right, you're next.
Deepa writes, Dear TWiV, it's sunny, windy, and 69 degrees in Hillsborough, California. You spoke about FIP, that's feline infectious peritonitis, a coronavirus of cats. On podcast 626, I was reminded about this article I read in The Atlantic on black market antivirals for FIP. Every molecule has a story. I'm an immunoparasitologist and chemokine immunologist turned banker turned investor who is learning a lot about virology from your excellent podcast. Boy, there's a career path. And so I haven't.
This is a great article. It's a nice article in The Atlantic. A much hyped COVID-19 drug is almost identical to a black market cat cure. So apparently, back in China, cats were getting sick last year. There was this black market drug, which was basically a Gilead compound that they said will cure your cat. So this one woman paid thousands of dollars and she got some vials and she said that it helped her cat. And now there's a black market and it's very similar to remdesivir, basically. So it's a cool.
You're supposed to, you're not sticking your cats on an IV, right?
I don't think so. I think you're just injecting it. Yeah, just injected the liquid into her dying cats every day for months. The remarkable thing, given the nature of the transaction, is that the vials actually worked. One cat lived another year and the other made a full recovery. So, anyway, it's a cool story about drugs. And here we are scrambling.
So,
for them.
actually, I'm looking at this because I've made a kind of a study of this. This looks like the unphosphorylated version of remdesivir.
Right.
Remdesivir has a 5' on what looks kind of like the ribose in this nucleoside analog, has a 5' phosphate. And then, as I described it before, spinach hanging off of the phosphate, a bunch of other stuff that I think is supposed to protect it makes that easier to be taken up in cells.
That's right.
But as I understand it, if you try and give that orally, it winds up all sucked up by the liver and triphosphorylated and never gets to where you want it to go because once it's triphosphorylated, it's stuck in the liver.
Yeah.
But this thing doesn't even have the phosphate on it. So if you were to just inject that, I guess it could be taken up all over the place.
Yeah.
But then it's got to be phosphorylated. I don't know what the probability of that is because I think that's a problem. So there aren't any real clinical studies on this. This Atlantic article is just anecdote, right?
Yeah, pretty much. But it's interesting. I think, it does work. And well, this is anecdotal, frankly. Okay. So I'm not sure how it's working. But it apparently, if you take the anecdotes, it's working in cats. And so somehow it's getting triphosphorylated, which has to happen inside of a cell. It's going to have side effects if that's the case. It's not going to be terribly virus specific, right? That's the beauty of acyclovir. The first phosphate gets done by TK, the viral enzyme, which then makes it specific for virus-infected cells.
So if I'm going to go off the deep end here, okay? And maybe there's a listener who can correct me if I'm wrong. But this idea about whether or not the compound that you're administering, the nucleoside analog, contains a 5' phosphate or not, makes what I'm thinking is that first phosphorylation that puts on the equivalent of an alpha phosphate is done by enzymes that are pretty specific for the nucleoside. Mm-hmm. So if the nucleoside, I'm thinking, is too wonky, okay, that phosphorylation is not going to happen efficiently. So maybe in order to make a drug work, you have to chemically put that on and administer it as a phosphorylated drug. But then you got the problem of charge and getting taken up by the liver and et cetera. The second two phosphates are fairly nonspecific. I think those are fairly nonspecific host enzymes.
That's right.
And I'm going a lot on the herpes story for that. Is that acyclovir, as you point out, is not monophosphorylated by the cellular enzymes. It takes the herpes enzyme that is a little more flexible than the cellular enzymes. But once that's done, the cellular enzymes can put on the other couple of phosphates.
However, AZT is not phosphorylated at all, and it gets into cells and it's triphosphorylated and inhibits, HIV.
Right.
The first drug.
So my assumption there is that AZT is just a nucleoside that fortuitously, even though it's not a normal nucleoside, our cellular enzymes don't have any problem putting on the first phosphate.
Yeah. Now, these drugs that have this, what looks like, phosphate in some spinach, as you say, they're actually clever chemistry because usually even one phosphate prevents efficient uptake from cells into cells.
Because it's charged.
Right. But they're disguising it, as you said, so that it's masked, right?
Yeah.
And then, as you say, the second and third phosphorylations are much quicker.
Right. And once again here, I'm pretty naive, but I think the liver is a remarkable organ.
It is.
Because everything has to go through the liver. The liver is in charge. And it's just a champion biochemist. And it sees this and it triphosphorylates it and basically says, I get it, you're staying here, dude. Okay.
We also got an email about the same article from Mason, who sent it. And he says cat owners have been successfully treating FIP with black market compound GS441524, a Gilead patented compound very similar to remdesivir. Okay. Actually, we got a lot more emails about feline infectious peritonitis, which I haven't queued up, but we'll get to because we asked for information and people are sending it in.
Yeah, maybe we ought to, I'd be interested to see whether there's, how good the data are, if there are any, on this remdesivir-like drug in cats, because just out of an intellectual curiosity.
You're talking about this 441524?
Yeah. I mean, maybe cats phosphorylate this stuff. Maybe cats can phosphorylate the precursor better than humans can, or something like that. I don't know.
Well,
but maybe it just doesn't work. I don't know.
Here's a paper, antiviral treatment using GS441524 in cats with FIP, published this year. It's a trial. So let's see what they did here. 4 cases of FIP were treated with this analog for 12 weeks. Cats were monitored. All cats had a positive response. 3 cats were alive after a year, and 1 cat was euthanized after relapses. And that's a very small study, but there's no pharmacology as you would like. Yeah, I'm sure there's more. Okay, let's go on to Guillermo. Hello, TWiV's crew. I heard you making some speculations about Brazil, and I'd like to clarify some things. One, although Brazil's health system is not state-of-the-art, as some more developed countries, it's not that bad. It's based on the British system being free and universal. Two, the real reason Brazil's doing so bad is political. I know you avoid the subject as much as you can, but it's impossible to analyze Brazil's situation without politics. Our president, Jair Bolsonaro, denies science, doesn't care about the 40,000 plus deaths, and has put all his chips on hydroxychloroquine as a viable cure, which we now know has been withdrawn by the FDA for use in emergency use. Governors, on the other hand, tried to make lockdowns, but we've never had more than 60% people at home, and due to the lack of federal help, people started pressuring for reopening the commerce, even though the cases never stopped rising. About the vaccine, we do have the ability to make it through Fiocruz and Butantan Institute. Also, due to how bad we're doing, we've become the perfect testing place. So Oxford University and a Chinese company called Sinovac are starting to make phase 3 tests here, 2,000 for Oxford and 9,000 for Sinovac. In exchange, they'll give us the means to produce the vaccines in case things go well. That's interesting.
Yeah, and I do, we have, you and I, Vincent, have Brazilian friends.
Yeah.
Nassim and Clarissa, who we've featured on this show in the past. And from my conversations with them, yes, the political situation in Brazil is precarious at best. It's not a good situation. Just another we try and avoid politics, but politics has a lot to do with public health.
Yeah.
And it can be a problem. And I'm just looking up to make sure if they're talking about which they say that there's a Oxford and Sinovac doing trials there. So the Oxford is going to be the chimp ad, and Sinovac is an inactivated whole virus.
That's right.
Interesting.
He says, P.S. also in Brazil, we avoid the word favela nowadays, preferring the word comunidades, communities.
Oh, okay. That's good to know.
I've been to Brazil twice for TWiVs, and Kathy and I have a friend in common, Eurico.
Yep.
And I have a former person who worked in the lab for a while there. I know I met lots of people, and it's a wonderful place.
I've had a Brazilian connection, mostly initially through Nassim for decades.
Yeah.
Been there a couple of times. I guess favela has become the politically incorrect term. Mm-hmm. It had a lot of negative connotations with it, so I'll try and remember that. Comunidades.
I bet Dixon used that last time, right?
I did.
You did?
Yep.
Oh, okay.
I didn't understand that it had become a disfavored word, but I stand corrected.
All right.
All right. Jason writes, Dear TWiV team, I'm writing from moderately overcast Berkeley, California, my hometown, my birthplace, where it is currently 22 degrees C. That's 295.15 Kelvin. On TWiV 262, you mentioned the Politico article headlined, Suddenly Social Justice Matters More Than Social Distance. Right.
So that's 620, let's see.
He says 262.
Yeah, that's
626. Yeah, there you go. With regards to protests, Brianne Barker also mentioned that we can have socially distant mask-wearing protests. Both statements seem to confound two very different concepts of social distance and social distancing. And he links to two wiki pages that contrast the two. It is unfortunate that the request for physical separation between individuals to curb the spread of infection has been termed social distancing or socially distant. Social distance is a concept within social sciences describing a sociological phenomenon. The term social distance is many decades old and refers to the separation of groups within a society by class, race, and ethnicity, and other groupings. Prejudice and racism are a manifestation of social distance. This is good. Social distance does not necessarily mean physical separation. There is considerable social distance between the guests at a luxury hotel and the staff who clean the rooms can be physically very, very close, but are separated by stark societal divides. There's considerable social distance between immigrant taxi drivers and wealth individuals riding in the back seat only a couple of feet away. The Sociology 101 example of social distance is the caste system in India. Ingrained rules meant to prevent intermixing by inherited social status. Social justice is in no way something that social distance opposes. Social distancing is poorly and unfortunately named because it does not aim to increase social distance. It aims to increase physical distance regardless of the social distinctions. Why the term social distancing was chosen rather than the more accurate term physical distance, a term that I have heard Dr. Fauci use more often of late, is beyond me. Protests against systemic and institutional racism should not be socially distant, nor should it in any way encourage greater social distance. Part of the goals of such protests is to recognize the social barriers that currently separate us and to move towards removing them. Risk of COVID-19 may require that protests involve more physical distance. The distance does not and should not involve erecting social barriers. I apologize for sounding pedantic. No problem with this crowd. But I would hope that as scientists, you would appreciate the value in accurate definitions and the danger in allowing buzzwords to obscure purpose and intent. Social distancing fails this. Both as it needlessly redefines physical distance as something else and as it needlessly intersects with a concept with a rich history in academic studies. Please, I implore you, stop needlessly tossing socially in front of distant when you mean physically distant. We can maintain physical distance while breaking down the barriers of social distance that are in direct opposition to social justice. That is brilliant. And we have been charged, Vincent.
All right. Maybe we'll call this episode Physically Distant.
Oh, excellent.
You know,
excellent.
True. I didn't invent it. That's for sure. I just heard people saying it. But let's use physical distance.
Absolutely.
Maintain your physical distance.
I have heard others make this same complaint, and it's absolutely true. And this is a perfect example of why it's inappropriate.
Yep.
Okay, it's excellent, excellent.
And here on TWiV, we love to be pedantic. Why do you think I complained at the top that D whatever G is not a mutation?
So, how would you, without prejudice, how would you title that paper? Because you do want to refer to that amino acid change.
Well, if you wanted to say D26 whatever G, you can call it an alteration or an amino acid change. In fact, we just wrote a grant application where we talk about amino acid changes, and that's what we do, alterations. And my colleague, we did a collaborative with another lab, and they were fully on with it because they're yeast lab and they understand genetic terminology, right? But, people drop it, and I'm sure you have dropped it in the past. Before I wrote a textbook, I dropped amino acid mutations all the time, but, let's be pedantic.
You're going to have a hard time curing me of that one.
That's right.
Okay.
I'm not going to, you're an old man.
Okay.
I don't need to cure you.
I don't know. I could use, actually. So this is another soapbox of mine. I regard change as a real challenge. Mm-hmm. Okay. People say that it's harder to change when you get older and that kind of stuff. Nonsense. Okay. My brain still works. Okay. I can do this.
So is there anything like, this amino acid mutation and stuff that you feel strongly about that's misused?
Well, actually, the first thing that pops in mind is the word data.
Mm-hmm. Yeah.
Okay. Now, I'm told that formally data can be singular or plural.
Oh, yeah?
But I'm not buying it.
I thought it was always
And I noticed that you use it as the plural.
Yeah.
And I used to hammer on my lab all the time about that. But that's a pretty minor one. I'll have to, we'll keep it in mind. We'll come across something that gets me. These are mostly grammar things. What is it? When people say stuff like, all of my family are not sick. When they mean not all of my family are sick.
Yeah.
Okay. Once again, that's just grammar. But we'll come across some science one will push my buttons down the road, and I'll try and notice that.
So Dave also wrote about physical versus social distancing and similar idea. And I just want to read. He writes, impatience and lack of understanding of this element of human nature isn't going to help as we forestall while waiting for effective therapies or vaccines. Nothing that I have seen suggests that on average we are much better off as a nation than we were a month or so ago. I would agree with that. I mean, in terms of, we have fewer cases, but the virus is still there and capable of spread. The only thing I would say is that some of the vaccines are making good progress, and I think that's good.
Yeah. I've never, I still strongly suspect that people have it in their heads that when the caseload starts to go down in a different location, in a given location, it means that the virus is disappearing. I think my guess is that in most people's minds, they don't get it that the virus is there. All right.
Yeah, I agree.
And I think, yeah, sure, the caseload has gone down and that kind of stuff. But all this time, the virus is spreading, man. You take your foot off the brake and it's gonna blossom. Mm-hmm. Deal with it.
Yep. All right. Zach writes, hello, TWiV. I'm a nurse practitioner, Yakima, Washington, where
Yakima. I think that's correct.
Yakima, where our county sadly has the highest per capita rate of lab-confirmed infections on the West Coast. I think this is really funny. People don't even have to mention the virus anymore in their emails.
Right. Lab-confirmed infections. Yeah. Good point.
Thank you for the informative podcast over the past decade, which I admittedly took a sabbatical from several years until the recent pandemic. In episode 626, you mentioned antigen testing. And I wanted to forward this along. Our clinic is going to start using an antigen test this week from the Kedel Corporation. It gives a link to that. Results are currently taking up to 4 days with our current lab, so I'm excited about the possibility of 15-minute results, just hoping they'll be reliable. So this is looking for viral antigens using, antibodies that you make, so that capture the antigens. And so that person would have to have an active infection. This is not like a serology test where the antibodies remain for a long time after infection is over. This is going to tell you if there are viral antigens present, which would be happening if the virus is replicating.
And so they say this is a lateral flow fluorescent immunoassay. So this is going to be, I presume it requires no special equipment or anything, and it's going to work kind of like the flu dipstick test, right?
Yeah, I think so. Yeah.
Which would be great. I remember when as you say, the qualification is how good is it? And we'll find out. Yeah. But I'm sure that this is only the first of a bunch of these. We'll see a bunch of these.
For sure.
Great.
All right. You can take the next one.
Amy writes, in a recent TWiV, the topic of who would get the first production of the SARS-CoV-2 vaccine. Are there organizations working to push for fair and data-driven vaccine distribution? Who could set that? Thanks for all you do. I feel more confident because I can listen to you. So, who was it we were talking to who invoked the AIDS network as...
Yeah, it was Ralph, I think, right?
That was Ralph. That's right. Which struck me as really interesting. So the AIDS vaccine network is a huge, complex network of contacts that was set up to do trials for the AIDS vaccine. I mean, as I imagine, organizing a trial and recruiting people and getting vaccine distributed and getting it evaluated is an extraordinarily complex task. And the AIDS vaccine network is all set up to do that. Now that, I don't know, I mean, I'm sure that that's set up with the idea in mind of using a population that is likely to experience HIV infection. Mm. So that may not have the kind of balance that Amy here is looking for. I don't know. I personally don't know how people would be recruited or identified for these various trials, whether or not the balance that she seeks will be achieved.
I think she's talking about once it's approved and it's distributed, right?
How
do we make it fair so that
yes, distribution. You're right.
We avoid the situation in the movie Contagion where people are going crazy trying to get the vaccine, right? And they had a lottery. Now, this is a really important issue. People are talking about this, but I don't believe that there's any plan yet, Amy, and it's a problem. I agree, because you can imagine, as we've said, countries with money will get it, and countries that don't will have to wait in line, and it's just not fair.
I think Ralph or someone was talking about this as well. It must have been Ralph, where the idea is, to try and get the vaccine where it's needed most and would do the most good to start with. You start off, one of the first categories is healthcare workers. Mm-hmm. All right. But also various at-risk populations, and that includes socioeconomically deprived communities, people with comorbidities, socioeconomically deprived nations. I don't know who might be supervising that, if anyone. Yeah. Good question.
All right, now we have a couple of emails, which I put in the category called, "Is Russia the New Sweden?" Not because of any political reason, but I don't know if you remember, listeners, when we got one email about how the outbreak was being handled in Sweden, and suddenly a flood of them afterwards. And the same thing is happening now with Russia, where we complained that for a while all the cases appeared to be in Moscow in Russia, which isn't correct, but that's how it was reported. And now we have some responses. So Angelina writes, "I wanted to say thank you for reading my comments about the COVID-19 situation in Russia. It's really great to hear. You're absolutely right that for the longest time, the Hopkins dashboard had one single dot on Russia, making it impossible to understand the extent of spread in the country. I noticed they only recently changed this, so now it's posted for different cities. Maybe they did it in response to your comments on TWiV. I hope so, as it was high time."
Putin is listening to TWiV?
I don't think so, but she's correct. It is fixed now. I also forwarded the episode with Chumakov to a History of Science network where it was appreciated and enjoyed. Now, if only I can come up with a way to make TWiV required listening for the students in my undergraduate art history courses. I'm sure there's a way with a little creativity. And Angelina is a professor of art history at a university, National Research University in Moscow.
Isn't that wonderful?
Cool.
Yeah.
All right, you can try and wade through this one if you want. Man. You don't see if you can pick out places. It's kind of long.
Yeah, it is kind of long. I'm really not very good at editing on the fly. I'll give it a rip. So worried about Russia writes, and this individual, who is who is anonymous in all ways, including sex or gender, and which I now know are two different things. I'm writing from St. Petersburg, Russia, where it's plus 13 and cloudy. He has some natural science background. As a recent listener, been retelling of facts about the origin of the virus and explaining the impossibility of its origin from a lab to acquaintances, advertising a podcast. Wanted to write complaining about people not washing their hands in public toilets, procrastinated. Then who declared a pandemic, quickly realized more important matters to discuss. Wants to write in response to the Muscovite letter, I guess the one that from the person we just read. Foreigners visiting Moscow often think the rest of Russia is more or less similar to the capital. However, Russia is better described as two countries in one, Moscow and all the rest. The difference between Moscow and the worst half of Russia proper is not something like New York, Mississippi, but rather New York versus Puerto Rico. Interesting. I don't know. Well, I won't go there. Moscow has a broad range testing scheme, but Russia proper has a shortage of tests. Regarding statistics of confirmed cases, thanks to the math, there can be no doubt that it's intentionally tampered with. The best is the so-called Krasnodar Krai Plateau. So where regional authorities reported that numbers of new diagnoses sort of plateaued off, even a schoolboy doing D in math would understand that these numbers are shamelessly faked. Not just one reason, up to half of Russia properly. So he's complaining that there is fiddling of the statistics in Russia. Lots been said in the Western press about statistics of death. The Russian Ministry of Health basically instructed forensic pathologists not to count death as caused by COVID-19 if an immediate cause of death allows it to describe death to something else, okay, stroke or etc. So the deaths are not being counted correctly. Guess is that many TWiV listeners are physicians. They might be interested to know that Russian medics are battling with the pandemic. In brief, Russian health professionals wage a war on two fronts, not just against the disease, but also their own superiors, because health administrators often treat them like dogs, depriving them of PPE, etc. And links to a couple of articles about this. Taken together with cover-ups of death, the ratio of medics who died from COVID-19 to the official death number is about 16 times higher in Russia than in other countries. Sorry, the letter's already too long. Well, I shortened it for you. I could write another one criticizing a Russian analog of CDC if you would like, but decided to skip that from now. But I can't refrain from briefly mentioning that despite yesterday saw the highest official number of new diagnoses in Russia proper ever, in less than 3 weeks, we are holding a national vote on a batch of 206 constitutional amendments, including one allowing Putin to re-elect until 2036. Already too strict restrictions are being lifted. So now I understand why he says, hence my pseudonym. Yours sincerely, worried about Russia.
Wow.
Yeah. Well, that is, we'll post the whole letter that you can read all the details on the website. But he describes a difficult situation in Russia that includes underreporting of disease, difficulties with testing, underreporting of deaths, even falsification of data, and manipulation of the political system.
So the links he provides are from the Moscow Times, and he says it's a great newspaper which I can recommend as a reliable source. They lack reporters outside Moscow. Oh, so the links are to something else, Meduza. He said this is a Latvian-based media. Meduza is better in this regard because they have reporters outside of Moscow, but he said the Moscow Times is also good. I can imagine that you could have good journalism in
I was
Russia,
thinking the same
right?
thing. How do you do reporting under these circumstances?
I mean, look at this country where it's all screwed up and we have supposedly a free press, right? Anyway, thank you. Worried about Russia.
Yes, thank you.
And then Ann writes, I heard you say in Sunday's TWiV that corona cases are only shown in Moscow. And then she sends a link to a website that shows the numbers by the region. This is region, that's Meduza. And this is a news site that reports on Russia from outside of Russia to avoid censorship. There you go.
Interesting.
Okay, here's another long one. I'll take this for you, okay?
Yeah, do that.
This is from Sheila. I've been a listener for only a few weeks, but an avid obsessed one. She's got taken my courses. She's listened to TWiV, TWIM, and Immune. Full-time mom with 3 kids and 3 dogs, no background in science, but a strange fascination with human immunity and microbes. One listener email said, listening to your pods had taken his knowledge to a higher level, but I think it's far more appropriate to say you have taken my knowledge to a lower level, meaning smaller. I am intrigued with the low-level microbial behavior. I have gotten into this stuff so that even my immunology PhD neighbor, who is in drug development at GSK, is sick of my questions.
Good.
Okay, questions. Through the pods and lectures, I learned of the distinction between infectious and non-infectious viral particles. I've not been able to find anything on the particle to PFU ratio for SARS-CoV-2. Great question.
Great question.
And that's what we were talking about up at the top. You know, why is the plaque assay not measuring infectious virus unless there's so much RNA around? Maybe there's a lot of defective particles. And she asks, if you inhale non-infectious particles, does your immune system mount an immune response? Great question. That's exactly what many people believe non-infectious or defective particles do. They stimulate the immune system to give you a better response. Being looked into. Whether it happens for SARS-CoV-2, we don't know.
By the way, I have a pedantic gripe about, now I don't think this qualifies as a gripe as serious as mutation. This is more an intellectual thing about particle to PFU. Because in order to measure the particle to PFU ratio for any given virus, you have to purify the virus. And you don't know how much damage you've done in the process.
That's right.
Now, in some cases, it's obvious because you do purify a preparation and you can see that there are a number of particles that are actually empty, don't have a genome in them. But not all viruses behave that way. And so, to me, particle to PFU, when measured, is a sort of a qualified number because you don't know how bad you've hurt it in the process of purifying it.
Yeah, I agree. Good point. All right, her second question. I've heard on some of the Immune podcast, guests express skepticism about vaccine safety on an accelerated timeline. I'm not an anti-vaxxer. I'm up to date on my shots as my kids are. If I had been a mother with young kids when Salk came out, I would have jumped on it. However, I have an apprehension about the safety and efficacy of a vaccine brought to market on an accelerated timeline with tremendous political pressure on manufacturers. I feel like going commercial will really be an extended phase 3 and would rather wait and see what happens to others who are more trusting. What are your thoughts?
She has learned well.
It's a great point. You're absolutely right. I don't like the accelerated timeline. I think having a disastrous vaccine is worse than COVID-19 in many ways because many people will now not take the vaccine.
Yep.
You cannot afford. Here's an example. In the 60s, a respiratory syncytial vaccine was developed. It was given to kids. The kids got worse disease as a consequence. And it took many years to figure out why. They figured it out. but as someone told me who worked on it, it took 50 years before we could convince people to think about making another vaccine against that virus. That's the price you pay when you make a mistake. And so we are going to be following it here. We'll see the results of the trials. We'll let you know what we think. But I agree.
And one of the biggest, I agree completely. One of the biggest concerns, in fact, is that very same thing that happened with the RSV vaccine, a vaccine-enhanced disease. And at very least, we can say that there are a lot of people on these problems. And I think most of these individuals are, if not all, I think there's a sufficiency of people concerned about this so that they will be evaluated as best they can be. But on an accelerated timeline, I can imagine where there might be issues. I mean, under the best of circumstances, some vaccines you don't find out what problems might be there until they really are put out in the population on a large scale, because there are rare enough complications so that you don't see them until those circumstances. But the other thing I worry about, as she points out here, is political pressure to get a vaccine or even a particular vaccine manufactured. And we have to resist that.
It's not like you can, take your pick of a bunch of vaccines, try one, try another. Once you have one, then the others might not work, right? I mean, if you get antibodies from Chadox against the spike, then maybe another one won't boost you as well. Who knows? So you're really going to have to go with whatever is offered to you. So, but we'll try and cover them all. Got time. And her last question is, I live in Santa Clara County, sunny 20C with annoying winds, which at the beginning of the outbreak appeared to be a hotspot. It appears that our early lockdown was an effective mitigation measure. Things are now opening up. Most people wear masks in public. Good. We are an environmentally conscious population. Many people bring their own reusable bags to grocery stores. However, county ordinance now forbids this. Isn't it now known that the risk of transmission by fomites is low, and doesn't wearing your own clothes and bringing your own purse and children present the same risk or possibly more as using your own grocery bags? I'd like to clarify, we are not even permitted to bring our bags into the store. It's not a question of making a cashier touch them. You'll be sent back to your car to put personal grocery bags back, or ask not to pull packable ones out of your purse, or the grocery store will be fined. I haven't heard of that before. Had you?
That's heavy. Well, yes. This is anecdotal because I think we wanted to go to a store at some point, and I asked my wife if she had her Trader Joe's bags along, because, of course, our reusable bags are all Trader Joe's bags.
Yeah. Yep. Me too.
And she said we can't bring them into the store. I forget what store it was. That we have to use their bags. I think she's got a point here. I think we know enough now so that the only possible risk that I can see is if you give the bags to the checker.
Yeah.
And they're packing your bag. But if you're packing your own bag, it ought to be okay. The thing that really annoyed me, we've been doing curbside pickup. The thing that really annoyed me is that the bagging in the curbside pickup was all plastic bags, and there were thousands of them. Some bags had just one item in them and that kind of stuff. But we've since found another branch of the same store that does it all in paper bags, and that's I'm more comfortable with that because we can recycle the paper bags.
Yeah,
I mean, you can recycle the plastic bags too, but to me, that's not. I'd rather get rid of plastic altogether as much as we could.
Yeah, I would agree. I think that contact transmission is low risk compared to airborne, right? And I think if you don't have anyone else touch the bags, it's fine, but they're not comfortable with it. So they're calling the shots. I don't want them to get fined.
Right. And I don't mind people being careful as long as it's not a great inconvenience.
Yeah. Thank you so for the awesome work you do. If I lived on the East Coast, I would volunteer to be an unpaid lab assistant and do your grunt work for you just because I find it so utterly fascinating. And Sheila, with those kind of questions, you'd be welcome.
Yeah. You wouldn't be doing grunt work for long.
No.
First thing I'd do after she washed all the dishes would be to teach Sheila to do a plaque assay.
Oh, yeah.
You'd love it.
She would. You're next.
By the way, I got to hang on.
Yeah.
See, get it up here.
You got a plaque. Is that vaccinia?
Yeah.
Oh, you brought one home.
How many people do you know that's got a plaque essay in their desk drawer? You probably do.
I do,
somewhere.
but yeah, that's beautiful. Those are lovely plaques.
You bet. That's one of the, I got lucky because when I started working on vaccinia, I didn't really understand that it was so good at this, but it makes just awesome plaques. Yeah. So one of the few things I saved when I shut things down was this particular plaque essay. I keep it in the dark because that
it won't fade.
crystal violet is going to fade over time. And I've tried to restain these at various times and it doesn't work well.
Of course, in my office, I have the wall covered with plaque assays.
Yeah.
1,672 6-well plates.
There you go. It's still there, right?
It is, yeah.
All right, Sandy?
Yep.
Sandy writes, Dear TWiV, I recently started listening over the last month. I'm grateful for your podcast. I'm a member of a Shevra Kaddish, Jewish ritual preparation for the deceased. Okay, this is good. The procedure is usually done within days of death. There is a great deal of water utilized during this process. We do suit up in full body protective gear, masks, face shields, and gloves. We have recently started doing Zoom services with the funeral home doing preparation of the body. I have the following questions. One, is there any data? I would say, are there any data on how long the viral particles remain infective or present on the body, nasopharyngeal areas after death? And two, I know this is a loaded question, but is it safe? Thanks for all you do, Sandy. I don't think there are any data for exactly the question you have, and I wouldn't want to do this. Doesn't sound safe to me.
Well, they're all suited up.
Yeah.
Full body protective gear, masks, face shields, gloves.
Yeah. Okay. If you're all suited up, I suppose you got plenty of healthcare practitioners who do this with people who are actively infected all the time, and they're okay. So if you're willing to take that, well, let's put it this way. I think there's significantly greater risk than zero, even though we don't have the numbers, that a person who dies of COVID-19, if you're preparing the body, is going to have some infectious virus on them for a period of time. Yeah. And so that's an exposure risk. So if you understand that and you're taking appropriate cautions, fine. But makes me a little nervous.
I would assume the virus is remaining infectious for at least a couple of days on a body.
I would assume so.
But whether it's enough to transmit, we don't know. So that's why it's a risk. And yes, I'm not aware of any studies that have looked at this. It's a very specific question. It's an interesting one.
I thought that, now I'm going to show my cultural naivete here, but I thought that in the strictest sense, the burial was supposed to take place within like 24 hours.
Oh, I don't know anything about it. I don't know anything about that.
Sure, I'm sure we'll get more mail.
There's one thing I do know is that George Floyd, he had COVID when he was living, and then they did during his autopsy. I think they did PCR, and he was still PCR positive.
Yep.
Which was over a month later, I believe. But that's PCR. It's not. We don't know if it's infectious virus. But when someone is deceased, the body stops to metabolize. Bacteria begin to grow, and you can imagine that the viruses are decomposing. There's no more alive cells to multiply in, right? So infectivity won't last forever, but And it also will depend on how long the person was infected, right? Because as we said in our earlier study, if it's over so many days, there's not likely to be infectious virus there since the onset of illness. Anyway.
Well, now this is a creepy question.
Mm-hmm.
When you die, how fast do all your cells actually cease metabolism?
Oh, I think they cease pretty quickly.
I guess they're not getting any oxygen.
Yeah. I mean, that's the big problem, right? They begin, they have oxygen. I think brain activity continues a little while, right? Firing and that sort of thing. But yeah, metabolism pretty quickly shuts down because there's no oxygen. Yeah. Let's ask the question, how long do cells live after death? It says here, 5% are alive after 70 hours.
Whoa.
After 3 days, the vast majority of cells are no longer viable. The last living cells would probably be commensal bacteria. Well, anyway, I think 5% of your cells alive after 70 hours. It could be that the ones on your skin could get some oxygen. I don't know. Maybe not. The outer layer is dead. I'm talking out of my wheelhouse here, but lately I've been doing that a lot. Have you?
Yeah, why not?
All right. Rupert writes, why do masks actually work? Dear TWiV, greetings from yet another newish listener. Thanks for everything. I really have a basic question. I know when someone sneezes or coughs, droplets spray out. Suppose someone with COVID-19 or a similar disease sneezes near me. I assume this means I'm going to be breathing in at least hundreds of particles with virus in them. If I'm wearing an N95, I guess that means around 5% will get into my system. That sounds like a lot. So how does a mask help me avoid getting sick? I can think of a couple of ideas. Maybe most virus particles get squashed by the immune system, so you need lots to get good odds of getting ill. Maybe the public health information videos about sneezing people were exaggerating, but would love an answer from someone who actually knows how this works. Rupert is from Cambridge, UK. Rupert, your first one nailed it. You need a lot of particles probably to initiate an infection. If you cut down 95% of them with a mask, it's probably not enough. And the mask, of course, is capturing the droplets, right? Not the virus particles, but the actual water droplets. And so I think that's how it works. It's a numbers game.
Yeah. It's repeated frequently that, yes, it's a numbers game, and getting infected is a function of both dose and time. So how much virus are you exposed to and over what period of time? And none of this is black and white. It's all gray. So wearing a mask is not going to 100% protect you, but it may cut down the dose that you get a little bit under whatever circumstances and reduce your risk to some extent. My understanding also has been for quite a while that one of the biggest benefits of masks is that it helps reduce spread from a person who is infected to others who are not.
Right.
So you're doing it. It's not just for you. It may be primarily for everybody else.
All right. Karen is next.
Karen writes, aloha. I've enjoyed your podcast. Aloha, by the way, is a really loaded word.
Is it?
It's a great word. It's more than just hello. It's got love in it.
Really?
Yep. I have enjoyed the podcast. I've been listening for the past few months. I've encouraged friends to listen as well, especially when they toot their horn without evidence. So, it's interesting. People who are sort of almost intrinsically or automatically sensitive to, wanting to see the data.
Mm-hmm.
For the areas that have zero or near zero cases, I believe the people of those areas are eliminating SARS-CoV-2.
I don't think so.
But let's go on. Hawaiian Island has had 0 cases for the past month. Hawaii Island. Hawaii's the big island, right?
Yeah.
No,
it's not.
Yes. Hawaii's the Oahu's where Honolulu is.
Yeah.
Hawaii's the big island.
Is it
Hawaii, and it's much less populated.
The island of Hawaii is the biggest island.
Yeah. Hawaii Island has had 0 cases for the past month. The protocol of testing, contact tracing, quarantine Tetris, along with masks and 6-foot social distancing, appears to have eliminated the virus from Hawaii, Maui, and Kauai Islands. Any new cases that occur in the future is because of people who travel to the mainland and return or travel in from the mainland. This is the case for Oahu. That's where Honolulu is. Hawaii state has the 3rd lowest number of cases of all U.S. states. If I understood correctly from the information discussed in your podcast, SARS-CoV-2 virus mutates, and it's unknown if a vaccine would be the answer. We need to address this. I heard discussion regarding revaccination periodically, and with SARS-CoV-2 virus antibodies does not mean that a person may not experience the symptoms again. Consequently, I see developing a vaccine is like shooting a moving target that replicates quickly. I see the way to rid ourselves is by eliminating through the practice of a protocol such as test, contact trace, quarantine, and wearing masks when 6 feet social distancing isn't possible, and social distancing, especially when not wearing a mask. The problem is that to eliminate this virus requires everyone being on board. I realize the islands and island counties have an easier time regulating and controlling incoming travelers. If residents in the USA would be cooperative versus protesting practices that may lead to eliminating a virus, USA would be in a better place. Please advise if my belief is off course and why. I listen to the podcast hoping to hear your response and discussion. If possible, when you do respond, let me know which podcast. Okay, and this is Karen, who lives on Hawaii Island. So, actually, I don't know what the disease burden on any of those islands ever was. Okay, so it strikes me as not completely out of the realm of possibility that those islands that she mentions, where there have been no cases, have little or even no virus on them. But in order to keep it that way, you would have to keep them completely isolated because the rest of the world has got it for good. It's not going away. There's too much, and it transmits too well, and it transmits in asymptomatic people. You would have to stay completely isolated for an indefinite period of time, and it's just not possible. The virus does mutate. There is no evidence whatsoever, as far as I know, that any changes in the genetic structure of the virus change its antigenicity. As far as I know, all the evidence is that it is antigenically stable, and so that any vaccine that we make should be good for the foreseeable future. And, this is based on not only our knowledge of SARS-CoV-2, but other coronaviruses as well. The coronaviruses do not undergo the same sort of antigenic drift and shift that, for example, the influenza viruses do. So we can make a vaccine to SARS-CoV-2, and a proper vaccine ought to be good for the foreseeable future. Now, there's another question in here. Oh, yeah, infection, reinfection. This is an unknown. Okay. Based on what we know about this virus and other coronaviruses, it is apparent that immunity, we don't know how long immunity lasts. There is good reason to believe that it may wane. There is good reason to believe that people may get reinfected. The hope is that the reinfections result in a much milder disease so that at equilibrium, some years out from here, it will behave as Ralph says. And Ralph talked about this the first time we had him on, and I was completely hooked. and this is where I've been ever since, that at equilibrium, several years out from this, okay, it'll be like a childhood disease. There'll be enough infection and reinfection in the adult population to maintain the virus, and there'll be infection of the younger population who don't get, by and large, a serious disease. The reinfections, hopefully, will give a much milder disease, and so this will be something that does not have the same sort of severity and consequences as it does in a naive population. Plus, I think it's quite possible we'll have a vaccine, okay? Years out, at least. So it's actually interesting to think that once this has become totally endemic and we've equilibrated with it.
You don't need a vaccine.
Yeah.
I think so. I totally agree with you. And all this effort and money, it will have been, I guess it will be useful to prevent a lot of deaths. But if it had, a fraction of it had been spent after SARS-1, we could have prevented this. Here are the numbers for Hawaii, by the way. 82 cases on the Big Island. Maui, 120 cases, 6 deaths. No deaths on the Big Island. Honolulu, 500 cases, 11 deaths. And Kauai, 21 cases and no deaths. So not a lot. And I would say if I tested every person on all those islands, I'll bet I would find infection somewhere.
Yeah.
The virus is still there. And it will always be there because unless you're going to eliminate people coming and going, they're going to bring it from elsewhere. And, New Zealand has shut down, right? Nobody can come in, but they can't stay that way forever. You can't. At some point, you have to ship stuff in that you need that you can't get locally. And that's going to bring virus in. And that's why we need high population immunity.
What I think about is somewhere along the line, this probably started with one person infected, right? And it's global.
I know. Isn't that awesome? Isn't that amazing? Not awesome. Don't get me wrong, folks. I'm not admiring it, but on a biological level, it's astounding, right?
Yeah. And given the virus, the global virus load on the population, you may try and keep it out, but it's going to sneak in unless you are completely and totally isolated and self-contained. Yeah. It's going to sneak in there.
Okay, Eric writes, listening to episode released on the 23rd, a comment was made not to get political on a science podcast. I found this comment ludicrous prima facie. It also flies in the face of good science and expert opinion. Folks, I'm not saying this. This is a listener. For example, to somehow ignore decades of social and economic injustice, creating swaths of our society that have larger and more pronounced comorbidities because it touches the no politics zone, does a disservice to listeners. Luckily, this comment specifically references a time you did not do that. The ability for listeners to be open to information that flies in the face of their understanding of the world crafted by whatever reality bubble they inhabit should be the price of admission, not the feedback in the complaints box. Keep doing what you do. Keep expressing yourselves in whatever method you deem necessary. Myself and others, I assume, realize you are humans living in a society and can make mistakes. Your heart is in the right place. Thanks for giving the release valve to anxious nerds like me who need to use knowledge to offset stress. Eric is from Calgary.
That's one of the best letters we've ever had. That is awesome. Very well said. I really like that. By the way, I'm sorry we didn't remember it when we talked about it last, but we mentioned Eric, who used to be the bat guy. It's Eric Donaldson.
Donaldson, right. Okay, sorry, Eric.
And I don't know where he is now. Eric, ping us. I believe it's Donaldson. It's not Davidson.
Yeah, I think you're right. All right, Gary's next.
Gary writes, hello to you all, and really enjoying TWiV. The insertion of 12 nucleotides into CoV-2 introduces a furin site, which you have discussed before on TWiV. The 4 amino acid insertion includes 2 adjacent arginines. which are both coded for by a CGG. This has the lowest codon usage bias of the 6 found in COVID-2, SARS, and MERS. Is this telling us something? Also, doesn't this argue against lab-made COVID-2 hypothesis? Wouldn't a researcher more likely choose AGA as the codon to insert? Just a thought, Gary. I thought he was going the other direction. But yeah, if I were going to create it in the lab, I would use the most frequently used codon. I don't know. The CGG doesn't tell me anything. But then again, I'm not a codon nerd.
Depends on the usage. It can be different in humans and bats, right?
Good point.
So I don't know. He says it has the lowest usage of arch codons, but I don't know if that's in human. If it's in humans,
he's comparing COVID-2, SARS, and MERS.
Yeah, so probably. I see. So okay, so they're probably talking about human codon usage. Yeah, I think that's a good point. You wouldn't use a rare codon most likely. So I think this is a good point to make. Sure, I'll buy it.
CGG, man.
CGG.
I'm seeing a compression on a sequencing gel.
Yep, it is. That's for us old guys who used to run gels. Yep.
Yeah, you gotta be an old guy to appreciate that one.
Gorka writes, hello, I'm a professor of telecommunication engineering in the Rey Juan Carlos University in Madrid, Spain, and an avid TWiVer, hopefully not in this sense. And apparently, according to the Urban Dictionary, TWiVer is a combination of twitching and shivering. I didn't know that, but in our book, TWiVer is someone who listens to TWiV. A couple of chapters ago, you mentioned PPV, positive predictive value, and NPV, negative predictive value, and their interpretation. These are, of course, for diagnostic tests. They are very important for public policy because with a low prevalence of the disease, even good tests, and most of the ones which we can get in Spain, and I suspect in other parts of the world aren't, have a high degree of uncertainty. A positive in normal circumstances with a commercial test, 5% prevalence of the disease, 95% specificity, 80% sensibility, I guess he means sensitivity, right?
Yeah.
Can convey only a 50% probability of being a true positive, which most people find completely counterintuitive. Even with better tests and a very low prevalence of disease, the effect of this effect of compounded uncertainty appears. People's eyes glaze over when you talk about the mathematics involved, but in this case, they're very important and not so difficult to understand. I've written a blog post in English and Spanish with this example worked out in simple diagrams to try and make it more understandable. In any case, thanks for your own down-to-earth explanations. And his name, Gorka, is pronounced like pickled cucumber in Swedish or like hill in Russian.
So is that Gurkha?
Gurkha? I don't know. Gurkha. Kathy would do it for us, but she's not here. And these are very good blog posts. Thank you very much.
Yeah, I'm going to have to read these because I fully confess that not only do my eyes glaze over, my whole brain glazes over.
It's a good explanation. It's very good.
Yeah, I'll have a look.
Elizabeth.
Elizabeth wants to know, hi there, any truth to blood type being associated with COVID and degree of illness, holding out hope for any reason?
And the next one from Aaron is the same question. And I put up a study from Chemical and Engineering News, an article. Genetic studies suggest that people's blood type may affect COVID-19 risk. So people with type A have a higher risk of severe illness, while type O may have lower risk. It's a genetic study of 1,600 patients in Italy and Spain that led to this conclusion. It's a medRxiv paper. So what does this mean? Well, there's an association, right? It's not a causation like most of these observational studies. And, what does it mean? And as we said earlier, we don't know how it would influence. You can speculate on what the mechanism is. But as I said earlier, next to the blood group locus is other genes that are involved in immune responses. So if you want to know more, it's possible that it could make a difference. The mechanism is unknown. I don't know what else.
I'm glad to see this. I was not, as I said, maybe this was in pre-show, or maybe we were talking about it before. I've seen this floating around, but I hadn't seen the study, and it's good to have this.
Yeah, there's a MedArchive article is linked there. So in this study, the researchers found they sequenced the genomes, right, of people with severe and not severe disease, or say severe disease and healthy subject. They found two regions of DNA in which the sequence variations were significantly tied to severity. One of the regions contains the gene encoding for the blood type, and but the other region contains immune genes. So, and they're close together. So, it's not clear what's going on here. But it could be that blood type controls disease severity. That has to be sorted out. And one of the ideas is that, blood type affects clotting, which can involve, and the disease can involve clotting, so that may be related to that. People with type O blood have lower levels of proteins that promote clotting, right? So they have less severe disease. So it makes sense, but it still needs to be proven, I think. All right, let's wrap it up. We got through some.
Yeah, that's 16 out of 152 pages.
The next one is from a UF alum.
Huh.
That's pretty funny. Yeah, we'll save it for another time. All right, that's TWiV. What number are we up to? 628. And you can find the show notes at microbe.tv/TWiV if you want to read the text of the letters. Some of them we didn't read all, and they're very good. We appreciate them. Microbe.tv/TWiV, you'll see a list of links, and at the bottom there is for the letters. You can send questions and comments to TWiV at microbe.tv. And by the time we have vaccines, hopefully we'll have gotten through all these questions about SARS-CoV-2. It's my goal. If you like what we do, consider supporting us. We could use your financial help. Microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, this is great fun. Always a good time.
I really appreciate it. I like to have at least one person here to think with.
It'd be hard to do alone.
It's back and forth. That's the whole thing. Yeah. That's science, right?
It's the whole idea. Conversation.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.